Welcome everyone to our third webinar of our Immunoassay Applications Week series, where we have live workshops every day going over best practices for different immunoassay applications. So my name is Dr. Poppy Nathan. I'm a scientific marketing specialist here at Proteintech, and I'll be chairing this workshop. And I'm really excited to have Dr. Amy Turner with us today to discuss all things Western blot. So we will have a Q&A session at the end, and I can answer some questions throughout in the chat. So please put any questions that you do have in the comments or in the Q&A box, and then we can come back to those at the end. But just before I hand over to Amy, I'd like to give you all a brief introduction to Proteintech in case you are unfamiliar with us as a company. So Proteintech is a leading life science reagent manufacturer. We were founded in 2001, and at that time we were a primary supplier of antibodies. We now have antibodies against over 14,000 human proteins, which covers about 2/3 of the total human proteome. We also offer immunoassay kits, including ELISA kits, IHC kits, cell separation kits, and antibody labeling kits. But since then, we've massively expanded our portfolio, and we've now got several more product lines. We have our humankind branded range of recombinant cytokines and growth factors, and all of our recombinant proteins are expressed in HEC293 cells, so you can be confident that they'll have the native folding, post-translational modifications, and glycosylation that you would expect to see in human proteins, which ultimately increases their bioactivity compared to proteins that are raised in something like an E. coli system. Importantly, we can manufacture all of our recombinant proteins at a GMP grade for use in cell and gene line therapies. We also have our Proteintech branded nanobody-based reagents, and nanobodies are actually very interesting because they're about 10 times smaller than traditional antibodies, and they combine to their targets with extremely high affinity, making them ideal for things like immunoprecipitation, super resolution microscopy, and protein purification. And finally, we have a subsidiary of Proteintech called Proteintech Genomics, which manufactures antibodies conjugated to oligonucleotides, which allows you to perform protein quantification alongside your RNA single-cell sequencing. So all of our award-winning products are self-manufactured. We have the largest portfolio of antibodies for the human proteome, and we have a rapidly expanding portfolio of recombinant antibodies. For convenience and testing, we provide most of these antibodies at a 20 microliter trial size for convenience and testing. We fully understand the importance of high quality product validation. So whenever we test new products, we validate with relevant sample types, appropriate experimental controls, and we validate using knockout and knockdown models wherever possible. All of our validation data is open access to our customers and available on our website. And we're a company of scientists, so we have an in-depth knowledge of immunoassay applications, and we can offer outstanding technical support for all of our customers. We have 7 inventory sites and manufacturing and research sites on 3 continents, which means we can offer next-day delivery to many countries across the globe. We always like to highlight that we define our success as a company through our customers' scientific success, specifically by helping you guys publish impactful research faster. We actually have over 350,000 citations globally, which makes us one of the top 4 cited antibody companies. And I also just want to highlight that as a company, we strive to make the searchability of our products as user-friendly and informative as possible. We've launched some new validation initiatives to help you guys select the right products. So we've experimentally mapped our recombinant antibodies, and we're sharing that epitope information with you through our unique 3D epitope viewer. So this allows you to interact with epitope structures and explore binding sites so you know exactly where our antibodies bind. We've also introduced advanced filter options to help you find exactly what you need, including our immunogen selection function. So this allows you to filter for antibodies generated specifically from your region of interest. And additionally, you no longer have to filter through all of the variations of an antibody from the search results. You can view all formats, including conjugations, buffer formulations on just one page, which ultimately is here to help streamline the process for antibody selection for our customers. And lastly, I want to highlight that we've built off of the information from our epitope mapper and database, and we've recently launched an AI-powered search tool on our website called Able AI. Able allows you to directly chat and ask for product recommendations, help for experimental design, and access relevant validation data, citations, and resources. For example, you can ask Able, recommend me specific antibodies validated for Western blot in this specific sample type, And Able will be able to suggest the best products for your experimental needs so that you don't have to spend countless hours researching for the right antibody. Able is a first-of-its-kind tool designed by our scientists in-house with customers in mind, so you can focus on our discoveries and not on reagent selection. We're really proud to be the first in the industry to launch this, so do check it out if you have time, either through the QR code on the screen there, or you can find it on our website homepage. But as I said, I'm really excited to be joined with Amy today. So Amy attained her PhD in structural and molecular biology from the University of Leeds, which focused on developing utility of Affimer reagents, which are a non-antibody binding protein, in streamlining the drug discovery pipeline, specifically targeting cancer-associated proteins, namely KRAS and PAC5. So leveraging her experience in structural biology and the cancer field, Amy transitioned from academia to industry and now works as a scientific liaison here at Proteintech, where she leads the European Field Marketing Representative Scheme, providing postgraduate researchers with industry acumen alongside their studies. So as a scientific liaison, Amy also delivers immunoassay workshops and creates content that resonates with Proteintech's diverse research audiences, including things like our detailed product guides, blog posts, protocol videos, webinars, and the famous Proteintech podcast. If you're not familiar with that, you can again find that all on our homepage. With that, just want to say a huge thank you, Amy, for being here and sharing all of your Western blot expertise with our audience. I'll stop sharing my screen now, let you take over. And as I mentioned, please feel free all to pop any questions you have for Amy just in the chat.
Thanks, Poppy. What a lovely introduction. So I'll go ahead and share my screen now. If you let me know that that has worked.
Yeah,
is that all good to go?
You can see that.
Perfect. All right, guys. Well, thank you so much for joining me today. There seems to like a lot of people in the chat all saying hello. So hello back to you. As Poppy has already introduced, I will be giving the third installment of our immunoassay applications week, namely on Western blotting. And today I'm going to be introducing a few little tips and tricks to accelerate your workflow. So if you've already done a Western blot before, or you've not done a Western blot before, you'll probably know that they take quite a long time. So I'm going to introduce a few new products, a few innovative developments from Proteintech that can help you speed up the workflows and ultimately your research progression. So in terms of the next hour, hopefully get it under an hour, we'll go through step-by-step protocol considerations. So exactly what you need to be thinking about at each step of your protocol and why you need to be thinking about them. I'll then go through some advanced tips for Western blotting, namely for proteins that are phosphorylated, those proteins that are large and also small, and also at the end, hopefully go through some Q&As with some troubleshooting advice sprinkled in there as well. So firstly, what is the purpose of a Western blot? And I'd just like to preface this by saying that obviously Poppy has already mentioned I do have a PhD in molecular biology. I did about 1,000 Western blots during my PhD and I actually really like them, which is an unusual opinion. But the point of them is essentially to separate a molecular mixture. So for example, a cellular lysate. So it's a lysate full of different proteins, separate them out, identify and quantify specific proteins of interest for you then to go on to do your downstream analysis. So there's a few different ways in which you can use a Western blot, a few different reasons why you would use a Western blot. The first one is expression analysis. So, for example, I'm going to use KRAS as my example because it is my favorite protein. If I was to look at KRAS, it's a cancer-associated protein. If I was to want to know the expression of KRAS in lots of different cancer cell lines, I could take my cancer cell lines, I could lyse them up, run them down an STS page gel, perform a Western blot on them and see exactly how much RAS was in each cell line and compare them to each other using immunoblotting. You can also use a Western blot to analyze post-translational modifications. So for example, if I have a cell line and I stimulate it with EGF, that would set off cell proliferation pathways. You can have a look for phosphorylation analysis. So for example, using the MAP kinase pathway, when you stimulate a cell line with EGF, MAP kinase pathway gets activated. Lots of different proteins will then subsequently be phosphorylated. You can run a Western blot to see how much of your protein is phosphorylated. So using a phospho-specific antibody against a protein of interest, for example, ERK or AKT. You can also look at protein localization. So for example, taking a cell line, you can fractionate it into whole cell lysate, cytoplasmic lysate fraction or nuclear fraction, run all of the fractions down a Western blot, blot for your particular protein of interest and see in which location in the cell your protein most wholly resides. And you can also use a Western blot as a post-processing step for interaction analysis. So, for example, using immunoprecipitation, which is another immunoassay that potentially is being talked about this week. So, for example, pulling protein A out of your sample lysate, running it down a Western blot and blotting with antibody, blotting for protein B with a different antibody to see if protein A was able to pull out protein B from your molecular mixture. Now, this is not kind of an extensive list of why you would use a Western blot. There is kind of a multitude of different utilities, but these are kind of the main examples of where you would use a Western blot. So in terms of the whole workflow overview, this is a kind of very simple schematic on what you do. And over the next few slides, I'll go through each individual step in a bit more detail and let you know what you need to be optimizing at each step. So the first step is preparing your protein sample. So if we're going to use the example of a cell lysate, this would be solubilizing your cell lysate to make sure all of those proteins are soluble and accessible. You then run the cell lysate down an SDS page gel, so separating your proteins based on molecular weight. You then take the gel that's got your proteins nicely resolved in it, transfer that onto a membrane, which is a little bit more robust, so your proteins move out of the gel and onto the membrane. You then block your membrane in traditional protocols. I will be talking about why you might not need to block in later slides. You then add your primary antibody, which binds to your protein of interest. You then add your secondary antibody, which binds to your primary antibody, and it has a detection moiety on there so that you can ultimately detect your protein of interest with this antibody interaction. So that's the main workflow, and over the next few slides, I'll go through a little bit more detail on how you do these. So starting off with preparing your protein sample. So like I said, Western blotting is kind of ubiquitously used throughout labs, and there's multiple reasons why you would use it. And there's multiple sample types that you can use, you can do a Western blot on. So sample types can be cell lines, primary cells, tissues, even whole organisms. So for example, if you are working with Drosophila or other model organisms that are quite small, you can actually homogenize the whole tissue of the whole organism and run that down a Western blot too. I did a lot of structural biology during my PhD, so I worked a lot with recombinant proteins and purified proteins. You can also run Western blots on these to check the purity of them, and many more different sample types as well. Now, the main kind of aims of the lysate preparation, the sample preparation, are to solubilize your proteins by breaking down the intra and intermolecular interactions. So you take your cell, in this case, hopefully you can see my mouse. If we take a whole cell, you want to start degrading or solubilizing the cell membrane, getting down to the quaternary structure of your protein, which is the protein complexes, so protein-protein complexes, then down to your tertiary structure, which is your single protein on its own, down to the secondary structure, which is the pleated sheets, so beta sheets or alpha helices, and then all the way down to that primary structure of just the amino acid sequence, nicely linearized and soluble. Now, during this process, what you also want to avoid is protein aggregation and precipitation, which can ultimately lead to sample loss. So there's a bit of a seesaw mechanism. You need to balance solubilizing your proteins whilst keeping them happy enough not to aggregate out of solution. So the main way that you will kind of lyse your sample, if we're taking a cell lysis here, is using a lysis solution, so chemical lysis. You kind of scrape your cells out into a tube, combine it with your lysis solution and let it do its thing. But what I want to kind of highlight in this slide is the different components of your lysis solution. Now, I know when I was doing my research, quite often you'd walk into a lab, you get handed a protocol that has been used for millennia, and it's got instructions on how to make your buffers. And especially when you're new to the lab, you just sort of add the components of the buffer, and you don't really think about why you're putting each of the components into the solution. So what I want to do on this slide is just highlight each of the different components of the lysis solution, why you're putting them into your tube, why you're putting them onto your cells, so that if you come to needing to optimize your protocol, you can figure out what concentrations of each component you might need to change. So in the lysis solution, you will always have a buffer system. So this is typically like tris HCl. So a buffer system maintains the physiological pH of your solution. So if you think back to my previous slide, again, you want to keep your proteins, you want to solubilize your proteins, but keep them happy enough in the solution that they don't precipitate out. So maintaining a pH that is similar to what that protein was residing in within the cell is a good way to do this. So typically between pH 7 and pH 8. You then want to add salt ions. So salt ions will maintain the ionic strength of the solution, again, maintaining what that protein would have been experiencing in the cellular environment. It also helps to disrupt the cell membrane when you add salt in there. But what you need to be aware of is that sometimes too high a salt concentration can actually make you downstream. You can get smiling bands in your Western blot, so just be aware of that. The third thing that you add into your lysis solution is chaotropic agents and detergents. So these are things like urea or Triton X100. So what these do is they weaken the hydrophobicity of proteins to solubilize them. So detergents will typically have a hydrophobic and a hydrophobic element of them. Hydrophilic element of them, sorry. So the hydrophobic element will interact with your proteins, and the hydrophilic element will interact with the water in your solution. So that will help to solubilize your proteins and maintain them in solution. The fourth thing on this slide here is protease and phosphatase inhibitors. So this is one of the most important things that you need to include in your lysis solution. These are inhibitors of phosphatases and proteases, as the name suggests. Now, proteases and phosphatases are enzymes which are within your cellular environment. They're ubiquitously expressed within your cell, so they can be within the nucleus, they can be within the cytoplasm, they can be within mitochondria. And when you start breaking down the walls of all these organelles, they get kind of broken out. The proteases, the phosphatases then have access to all proteins within your sample. And what these enzymes do is they will chop up proteins, they will degrade your proteins, that is their use. But obviously, if you're wanting to detect your protein downstream, then you don't want it to be degraded. So you need to inhibit these with phosphatases and protease inhibitors. And then finally, you want to add a reducing agent, so either beta-mercaptoethanol or DTT. Now, this helps to disrupt the disulfide bonds that occur between two proteins or within a protein, so that you're fully denaturing down to that primary structure. Now, the thing that you need to think about as well is where your protein of interest resides within the cell. So, for example, if you've got a protein that lives in the nucleus, when you start degrading the cell membrane, so solubilizing that cell membrane, you will also have to solubilize the nuclear membrane in order to have access to your protein of interest, which means that you might actually need to use a stronger detergent than if somebody was looking at a cytoplasmic protein that just resides within the cytoplasm and you only have to break down one membrane. So just to those people who are new to the lab and new to the Western blot protocol, if you have been given a protocol by somebody else who is working on a different protein to you, make sure that you are tailoring the lysis solution to the protein that you are actually interested in and where it resides in the cell. So Proteintech's top tips for an effective sample lysis is, like I said, tailor your solution to your particular sample type and your protein of interest. If your final lysate looks really gloopy, you might have to do a sonication step. So this is using basically sonar waves to mechanistically break down your sample. So there's basically putting it in a sonicator, it adds sonar waves, and that will kind of break down any bonds that have been difficult to break down using chemical lysis. If you keep your samples on ice, this also decreases any enzyme activity from those phosphatases and proteases. So with most immunoassays, if you keep your samples on ice, it reduces any catalytic activity of things that are going to start to break down your proteins of interest before you have a chance to detect them. So that's our sort of top tips. Proteintech have also got lots of resources online, including a full webinar on how to sort of optimize your sample prep, your lysate prep. So you can go kind of offline and have a look at that webinar too. You can put it at double speed so you can get to the right section that's of interest to you too. So once you've got your lysate nicely soluble and all your proteins are nicely soluble, you then need to combine it with an STS sample buffer. So the STS sample buffer is what you need to essentially give those denatured proteins a negative charge, which is necessary for it to migrate down your STS page. So again, just looking at the components of the STS sample buffer. I won't do this for all the buffers, but I just want to get your kind of head around thinking about what's in your buffers. So the first thing that's in an STS sample buffer is, of course, STS. So this is the element that gives your proteins that negative charge. So I always think of my proteins, or I used to think of the proteins that I was working with as a piece of rope. And when they're in their native form, it's in a specific knot in a specific shape. But when you have denatured it, it's a long string. You then want to add this negative charge all the way along it so that when your proteins start to migrate down that STS page gel, they'll migrate purely based on their molecular weight rather than the shape of them or the charge of them. So they have this uniform negative charge. The other thing that's in this buffer is glycerol. So I'm sure I think there's about 500 people in this call right now. Many of you might have already done a Western blot. When you start running that STS page gel, or when you start loading the STS page gel, I'm sure that at least one of you has done it and your sample has floated out of the well. I did it many times. That's because the sample isn't dense enough to stay in the well. So that's the reason that we add glycerol into the STS sample buffer. That just increases the density and makes sure that the sample stays in the well so that then it will migrate when you turn it on. In your sample buffer as well, you have bromophenol blue. This is purely to make the sample blue so that you can assess its migration down your STS page. And then finally, you also add reducing agent into the STS sample buffer, again, just to make sure that any of those disulfide bonds are fully denatured and what you're getting is perfect migration of your proteins. So most protocols will tell you then to boil your samples. So when you've prepped your lysate, you've combined it with the sample buffer, they'll say boil your samples at 95 degrees for 5 to 10 minutes. And in most cases, this is appropriate. It means that you've got kind of complete denaturation of your sample. It really disrupts the hydrogen bonds, the hydrophobic interactions, and the van der Waals forces. And it makes sure that your protein's fully unfolded. However, if anybody is using a membrane protein, these typically are really big proteins and they are sticky proteins. They're quite notoriously difficult to work with. And occasionally heating up these proteins will mean that they actually crash out of solution. And if that happens, they can aggregate and clump together and not actually be able to migrate down your subsequent STS page gel. So if you are using a membrane protein and you are having issues with detection, so for example, in this gel here, what we can see is that this transporter, big membrane protein, you can see it's running at sort of 160 kilodaltons there. When we haven't boiled it, we could get really nice detection, really clean detection, but actually when we have boiled it, we can't see anything. And that's because it's stuck at the top or it's precipitated out of solution. So my tip for people who are working with membrane proteins who might be having issues with detection is actually do a temperature titration essentially on your samples. So once you've solubilized and once you've made your lysate, incubate at different temperatures, not just 95 degrees for 10 minutes. You can go colder than room temperature, all the way up to 100 degrees, and run those samples under the same conditions and see at which point you can actually see your sample most effectively. My kind of health and safety tip here is if you are using urea in your sample buffer as a chaotropic agent or your lysis solution as a chaotropic agent, don't heat above 37 degrees because it can actually form ammonia, which can then kind of interfere with the characterization of your protein. So if you do use urea, don't heat anything above 37 degrees. So hopefully at this stage, you've got a really nice protein sample that is combined with your STS sample buffer. The next thing to do is a STS page. And this, if most of you probably have done a biochemistry degree, the kind of thing that's ringing in my ear is that an STS page separates your proteins based on the size. And this is true. So an STS page uses a acrylamide gel to separate your proteins based on size. And the gel, I always think it's like a mesh. So it's not a solid medium, it's a mesh-like structure. So it has tiny holes in it where your proteins can migrate. But what that means is that if the gel has the thing that makes the mesh is called acrylamide, that's the component that makes your mesh. If you have a high percentage acrylamide, the holes in the mesh are smaller. If you have a lower percentage acrylamide, the pores in the mesh are a little bit larger. So this is important to know, and you need to use the percentage acrylamide that is most appropriate for the protein of interest that you're looking at. So if you're looking at a particularly large protein, so anywhere between 80 and 200 kilodaltons, then you're going to need a acrylamide gel whose pore size is large enough to allow those proteins to migrate effectively. If you're looking at a tiny protein, so anything from sort of 10 kilodaltons to 30, 40 kilodaltons, then you're going to want a higher percentage acrylamide gel, so one with smaller pores, so that those smaller proteins can migrate effectively and the larger proteins will kind of stay at the top. So smaller proteins migrate faster. They can fit through the pores a lot faster and therefore they travel farther down the gel. Higher molecular weight proteins take longer, so they end up higher up the gel. You can also use gradient gels, which have a gradient of acrylamide. So each section of the gel, it's got different percentage acrylamide, so the size of the pores is different. And this can be good if you are working with 2 or 3 different molecular weight proteins and you want to detect them at the same time. So our tips, if you are coming to a Western blot protocol for the first time with a new protein, is load at least 30 micrograms of your protein, especially if you don't know what the expression levels are. Use a suitable gel percentage based on molecular weight, like I said. And if you are seeing smiling bands, which if you've done a Western blot many times, you probably at some point have seen a band that is smiley. Might look nice, but it's not very effective for publications. What we suggest there is that you decrease the voltage that you are running in that SDS page gel. So there's always a tendency to run it at a higher voltage so that it goes faster and you can go for lunch or you can go home quicker. But actually what you want to do is run it slowly so that they resolve nicely and you don't get smiling bands. At this point as well, don't forget your molecular weight ladder and do be careful to use a protein molecular weight ladder. I many times use the DNA ladder, but this allows you to determine the molecular weight of the protein of interest that you're interested in, and it makes sure that you're actually validating. So it validates the antibody. So for example, if you're looking for a 50 kilodalton protein and the antibody detects something that is at 50, then you can kind of assume that the antibody is targeting the right thing. It's also really important to use a molecular weight ladder for assessing the gel migration. So if you've made your own gels, what you want to know is that it's consistent all the way through. You've got like consistent pore sizes and that the proteins will be nicely migrating. Usually molecular weight ladders are coloured so you can see them, and they're an indicator that the gel is run properly. And if they've kind of run all skew with, then you can safely assume that the gel composition might need optimizing. It's also good for checking the transfer efficiency. So when you transfer later on, the proteins move out of the gel and onto your membrane. If you can see that all of the coloured bands have moved onto the transfer membrane, then again, you can safely assume that you can move on with the protocol. So again, if you are coming to the Western blot for the very first time and you want to optimize really effectively all the way through, something that I recommend doing is actually doing a Kamasi blue stain after your SDS page. So when you run your SDS page, you'll have hopefully nicely resolved your proteins based on size, and you'll have a gel that has all of your sample lysate all nicely separated. But what you want to do at this stage is actually check that your lysate was effective and your gel composition is optimal for your particular protein of interest. So what you can do here is do a Kamasi blue stain, which is where you take your gel and incubate it in Kamasi blue. Kamasi blue binds to hydrophobic and basic amino acids, which are rife within most protein structures. And it gives you these really clear blue resolved bands, which indicate your proteins. And that should show up against a clear background of the gel. It's a really simple procedure, and what it shows you is essentially how effective you've been able to lyse your cell samples and how effectively you've solubilized your proteins. So what you want to see is this. So the sample on the right here, you can see really clear blue bands all the way along. What you don't want is a big clump of proteins at the top. So if you do have a big clump of proteins at the top, that's indicating that either your gel percentage is too high or your lysate isn't effective. So you need to go back to your right at the very beginning and optimize your cell lysate procedure. What I will say at this stage is that if you do decide to do a Comati Blue to check your lysate, you kind of can't then do the Western blot after it. You're going to have to run this in parallel because this is an irreversible step. But once you have optimized both your lysate and your STS page gel steps, you then go on to transfer. So like I said right at the beginning, the transfer is essentially moving those proteins that are in the gel now onto a membrane. So it's a little bit more of a robust medium that you can then use to immunoblot, so using antibodies to detect your protein of interest. And there are two main methods that people now use to do this. So wet transfer is the kind of traditional method. It's what was always used back in the day. Basically, you take your gel that you've resolved your proteins on, you put it next to the membrane, you sandwich that with blotting paper, and then put that into a cassette, which is then submerged in transfer buffer. So it's all within transfer buffer. You then add a charge to this, and those negative proteins will move out of the gel and onto the membrane. It takes about an hour to 2 hours or overnight, and it's really good for high molecular weight proteins and low abundant proteins because it's really, really efficient. So I think the active kind of submerging it all within that transfer buffer kind of helps to push those proteins out of the gel and onto the membrane. However, most people these days actually use semi-dry transfer methods, and this is because it's a little bit faster and a little bit more convenient. So the difference here is that the whole kind of sandwich stack of your gel, your membrane, and your blotting paper is not all submerged in transfer buffer. It is held between two electrode plates that you kind of put into a little cassette, and it takes between 5 minutes and 30 minutes to transfer all those proteins. So it's a little bit faster and usually a bit more convenient for people. It's suitable for a wide range of molecular weights as well. However, if you are struggling with the transfer efficiency, especially if you're using a really high molecular weight protein, then I would suggest going all the way back to wet transfer because it is just that bit more efficient. The other thing that you need to think about, and it's something that I never thought about when I was doing my PhD, is actually the membrane of choice. So there's two main membranes that you can choose from. You've got PVDF and you've got nitrocellulose. So I genuinely didn't think there was much of a difference, but actually there's a massive difference. And if this is the one thing that you take from this session, just remember it. So the biggest difference is actually the way that these membranes stick to your proteins. PVDF membranes will stick to your proteins using hydrophobic and dipole interactions. Nitrocellulose membranes will stick to your proteins using hydrophilic and electrostatic interactions. Now, you might think that doesn't matter as long as they're sticking to my proteins, but actually 80% of antibodies use hydrophilic and electrostatic interactions to bind to your protein. And if you've already masked those interaction sites using the membrane, then actually you can be kind of decreasing the signal that you might get from that antibody that will also use those sites. So my tip would be to use PVDF membranes if you are struggling with the signal strength of your ultimate detection. They have a higher sensitivity and they also bind to your, they've got a 2 times higher binding capacity than nitrocellulose, which means that they bind more protein. Something to think about as well here is that the membranes, much like the agarose gels, are also like a mesh. So again, they have pore sizes. So you need to think about the pore size that is most relevant for your protein of interest. So pore sizes can range from 0.2 microns to 0.45 microns. And again, you just need to make sure that if you're, for example, looking at a tiny protein, so 10 kilodaltons to 30 kilodaltons, you don't want to be using a pore size that is too big because that protein can actually go onto your membrane and out the other side. So make sure that you're using the right one for you. Another tip here is actually once you've done your transfer, your gel should be empty of protein. And there is a way that you can check that. You can go back to that Comati Blue staining that I talked about earlier. Very quick stain, incubate in the Comati Blue stain for maybe an hour, do a destain, and what you should hopefully see is that there's no proteins there. There's no detection of any of those blue bands, and that would indicate that you've had an effective transfer. Always remove the air bubbles as well. So if there's people in the audience, who've already done a Western blot before, you'll know that bubbles can appear when you make that stack. So making the stack of the membrane and the gel and the blotting paper, bubbles can occur between the gel and the membrane, and that will obviously affect your protein being able to migrate across. Usually what will happen is that bubble will occur right on the point where your protein of interest is. So make sure that you roll it out using a roller, and make sure that you can get an effective transfer across the whole of the membrane. So once you have transferred your proteins onto that membrane, what you then want to do is block the membrane. But again, I've got a little bit of a caveat here, but we will talk about that now. So blocking is a kind of universal step for most immunoassays. It's a way for you to prevent non-specific binding of antibodies. So if you imagine at this point, you've got a membrane, your cell lysate, whatever sample you're using, has transferred onto that membrane, and there will be parts on the membrane that are empty, essentially empty and ready for protein binding. And an antibody, which you'll later use to detect your protein of interest, is a protein. So it's charged and it will stick to that empty portion of that membrane non-specifically. So you want to avoid this by blocking the membrane, and blocking the membrane essentially adds non-specific proteins onto those empty portions of the membrane so that your subsequent antibody then won't bind. So typically you would use non-fat milk or bovine serum albumin, BSA, which are non-specific proteins that would then saturate that membrane and make sure that your antibody doesn't non-specifically bind. Within that blocking solution, you would also, the base of it would be TBST or PBST, and that element of T, the tween, is a detergent, and that just aids the wetting of the membrane, so it prevents it drying out. However, it does take about an hour to do this. So typical Western blot, traditional Western blot protocols will suggest that you take your membrane out of your transfer machine, give it a quick wash, and submerge it in blocking buffer for at least an hour. However, Proteintech have actually developed a new solution, which means that you don't need to block anymore. It's called our blotting accelerator. If you want any more information on it, please scan the QR code. But essentially, if you take your traditional blotting protocol, like I've just sort of been describing now, you do your STS page, you transfer, you then block for an hour, you then add your primary antibody for an hour, you then have to wash, you then have to do another incubation with a secondary antibody, then you do another wash, and then you can detect. So it does take quite a long time. So Proteintech, like I said, we have developed this blotting accelerator. It's essentially a buffer that you dilute your antibody in instead of milk or BSA. And what you do, you essentially skip out that blocking step and you go straight from transfer, very, very quick wash in TBST or PBST for 3 to 5 seconds. You then incubate with your primary antibody in the blotting accelerator, and you only need to incubate for up to 25 minutes. So 10 minutes to 25 minutes. You then again do a very quick wash, so 3 times 1 minute instead of 5 times 5 minutes. You then add your secondary antibody again in the blotting accelerator, and you can incubate from as little as 8 minutes to 15 minutes. Then do a quick wash, again 3 times 1 minute, and then you go on to do your detection. So it takes your protocol from 4 hours to 40 minutes, which is something that is kind of revolutionary. I've got a colleague in Rotterdam actually who did their Western blot post-transfer in 3 minutes, which is a bit of a record. So if you are interested in using this blotting accelerator, please do have a look on our website using that QR code. So going back to kind of the traditional Western blot, which most people will be doing at this stage, after you've blocked, you then incubate with your primary antibody, which is specific to your protein of interest. Now, choosing the right primary antibody is paramount. Obviously, you want to target your protein of interest. So I'm going to use KRAS as my example. When I was think back to when I was doing my PhD, I didn't really care about the clonality of the antibody, didn't really know the difference. I just sort of used what was in the freezer. But what I also want to make sure that everybody knows is that it's really highly important that you do understand the difference between the clones of antibodies, because it can affect optimization downstream if you are having issues with your detection of your protein. So I'm just going to briefly go through each of the clonalities and what their benefits are. So polyclonal antibodies are your kind of traditional, conventional antibody. This was the original kind of antibody reagent that people have used for years. These, as their name suggests, are multiple clones of antibodies within one mixture. So each of the antibodies in that one tube binds to the same protein, but it will bind at different sites on that protein. So multiple different epitopes on that protein will be covered by the polyclonal antibody. These are derived from the serum of immunized animals. So, for example, at Proteintech, the polyclonal antibodies are raised in rabbit. You immunize the rabbit with the antigen of interest. That rabbit will raise an immune response, and we take the antibodies from that immune response, and that's what your polyclonal antibody is. So it recognizes multiple epitopes, so it has high affinity, essentially. So the avidity effect of multiple antibodies binding to one protein means it's got high affinity. It's great for rare species that might have whose kind of the homology between for example, if you immunize the animal with a human protein and you're working on maybe a zebrafish protein equivalent, the kind of chance that there is a homologous region on the zebrafish protein compared to the human protein that is covered by one of the clones, at least one of the clones of the polyclonal antibody, is quite high with polyclonals. So it's great for rare species, and it's good for low abundant targets because you get that signal amplification from the multiple clones binding. There is also monoclonal antibodies, which, again, as the name suggests, are a singular clone. So they recognize one single epitope on your protein of interest. The way we produce these is we take the B cell of the animal that is producing antibodies against a particular protein. We fuse that B cell with a myeloma cell line, which is a cancer cell line, so it's highly proliferative. That forms a hybridoma, which means that you essentially internalize this B cell that is producing this antibody of interest. So then you just have a cell line that continually produces the antibody against a single epitope on your protein of interest. These have really low background because they, again, just only bind that one epitope. They're highly specific, and they have minimal lot-to-lot variability because they are being produced from that one cell line that is just continually proliferating. We then come on to recombinant antibodies. So this is the kind of shiny new antibody. It's been on the market for maybe the last 5 to 10 years. But these antibodies combine the high affinity of a polyclonal reagent with the high specificity of a monoclonal reagent. In this sense, we take essentially the DNA code of the heavy chain and the light chain of the antibodies. We put this into a cloning vector, and then we recombinantly produce these antibodies in human cells. So these recognize defined epitopes, and they're kind of engineered for specificity, and they have no lot-to-lot variability. So the reason they have no lot-to-lot variability is because we know their sequence, and we recombinantly produce them. So there's no genetic drift. There's no difference in sort of bleeds of different animals or the immune response. We have that genetic code. We know exactly each time that we're producing the exact same reagent. So they are perfect for long-term projects where you want really, really high reproducibility in your assays. So they're the kind of 3 main clones of antibody that you need to be thinking about. Choose based on the protein of interest, essentially, that you're working with. You also need to plan your controls. So as well as finding an antibody for your protein of interest, you also need to plan antibodies for control proteins. The kind of mothership of a control protein for a Western blot is your housekeeping proteins. So these are proteins that usually are involved in the structural parts of the cell or metabolic parts of the cell that will not change depending on kind of the conditions that you put your samples under. So these are things like GAPDH, beta-actin, beta-tubulin. The thing that I want to highlight here is pick a loading control based on where your protein of interest is in the cell. So most people, if you're doing like a whole cell, if your protein moves around the cell, it's nuclear, it's cytoplasmic, it's membrane, wherever, your kind of gold standards are your GAPDH, your beta-tubulin, and your actin. Your actin. So they're pretty ubiquitously used throughout most proteins. But if you're using a mitochondrial protein, if you're studying mitochondrial protein, you might consider using a loading control that is actually more suitable for mitochondria. So using maybe COX-4 or VDAC1, which are within the mitochondria and will sort of be more appropriate for your target of interest. You also want to use antigen controls. So if you were in a Frida's talk yesterday for the IF stuff, antigen controls are also really useful for immunofluorescence. But these are your positive and negative controls. So positive being a sample that overexpresses your protein of interest, and a negative control is a sample that does not express your protein of interest. So if you're wondering, where you can get hold of a sample that doesn't express your protein of interest, either you can make it yourself, so using an siRNA or an shRNA knockdown, or you can find commercially available cell lines or samples online using the PAXDB or the Human Protein Atlas. I've also put the Able logo up in the corner there. Able AI is our AI-powered search tool on Proteintech's website. It's fantastic for looking for the most appropriate controls for your research as well. On here as well, we've got phosphoprotein controls. So if you're looking at a phosphorylated protein, I always recommend looking at the whole total protein as well. So if you're looking at phospho AKT, for example, also look at AKT as a whole, so not just using the phospho-specific antibody. You should always do a no primary antibody control as well if you are in the kind of first stages of optimizing your protocol. So this is essentially, like it says on the tin, not using a primary antibody, just using a secondary antibody, and this will check that your secondary antibody isn't non-specifically binding to the membrane or to the sample, something in your sample. You also want to do an isotype control. Again, an isotype control if you're really wanting to optimize very, very efficiently at the start of a project or the start of an assay. Isotype controls are super important. An isotype control is essentially using an antibody that is from the same host. It has the same IgG class and subclass, but it does not bind to your protein of interest. So essentially, it is the exact same antibody that you have for your protein of interest, apart from the antigen binding domain. So apart from the bit that is specific for your protein of interest. And what this does is it makes sure that if you use this as a control, it makes sure that none of the backbone of the antibody is actually doing anything non-specific on your membrane. And if it is, then you can use a different antibody essentially. So I did actually want to share, like I mentioned, Able AI is super useful for planning your experiments. And if you've been to the other immunoassay workshops this week, I'm sure that they've also highlighted it. But I just wanted to give an example of how you could use it for Western blotting. And again, I've used my favorite protein KRAS. So what antibody combination should I use to best detect KRAS in Western blotting? That is a question that I asked Able this morning. It came up with the KRAS specific antibody, came up with a pan-RAS antibody, and then recommended secondary antibodies for a combination assay there. So it's super useful for you to pick the initial antibodies right off the bat that have been validated for Western blot. And then I said, is there any more tips that you have for detecting RAS? And it's given me antibody selection. So it's delved down into the different isoforms of RAS, so 4A and 4B. And what it actually came up with here, which I didn't think of, is actually changing the blocking reagent, so from milk to BSA in this case. So even if you think that you know your protein of interest, just put it into Able and see if it comes up with something different, because it is super useful. So moving on from primary antibody controls, after you've done your primary antibody incubation, so typically if you're doing a traditional methodology, you will incubate your membrane for an hour with your primary antibody, unless you're using the blotting accelerator. You then give it a quick wash, so 5 times 5 minutes, and then add your secondary antibody to detect your primary with a detection moiety on there. So there's a few things that you need to think about. So choosing the right secondary reagent. If you're using an antibody, obviously you need to use a secondary antibody that can bind to the host species of your primary. So for example, if you've used a rabbit primary against RAS, you want an anti-rabbit secondary. The thing that I want to highlight here is if you're using a mouse primary, you can actually detect different subclasses of mouse primary antibodies with secondary antibodies that differentiate between the different IgG subclasses. So IgG1 and IgG2A, you can get secondary antibodies that bind them differentially. And then if you're using an antibody, you need to think about the detection moiety that you want to use to ultimately detect your protein of interest. So there's a couple of options. One is HRP, so horseradish peroxidase. This is a typical secondary antibody conjugation. It's an enzyme that when you add the luminol substrate to, it oxidizes it, and through that oxidization of the luminol releases light that can then be detected by either an X-ray film or a CCD camera. You can also use fluorescently labeled secondary antibodies, which can give you a little bit more ease of multiplexing, and you get a greater quantitative dynamic range, which essentially means you can quantify a little bit more accurately how much protein is on your membrane. If you don't want to use an antibody as a secondary reagent, there is also other options, especially at Proteintech. So we have nanosecondary reagents. So these are the nanobodies that Poppy mentioned right at the beginning. And then we also have Flexible 2.0, which I believe is on my next slide. Yes, it is. So Flexible 2.0, again, if you were in the imaging immunoassay talk yesterday, you might have already seen this, but essentially it is an antibody labeling kit, which will label your primary antibody with either a HRP enzyme or a fluorescent molecule, and it takes 10 minutes. So you take your primary antibody, it doesn't need to be from Proteintech, it can be anything that you've already optimized for your particular assay. You incubate it with the Flex linker. So again, that will add either a HRP or a fluorescent molecule, depending on what your detection moiety is. Incubate for 5 minutes. You then add a quencher, which will mop up the spare linker, and then you then put that antibody directly onto your sample, if it's IF, or your membrane, if it's Western blotting. So it literally takes 10 minutes. If you combine that with our blotting accelerator, and we do sell kits, so they're called SpeedAb Western blot kits, you can actually essentially do a one-step detection of your protein of interest, and it takes literally 2 hours, which compared to a traditional Western blot, which would take 5 hours, where you do, your primary antibody, you wash, your secondary antibody, it's a lot easier. So again, if you're interested in speeding up your Western blot, you can either just use the blotting accelerator, or you can use our kits, which combine the antibody labeling kit, which adds the HRP to your primary, and then your blotting accelerator, so that you can do it in 2 hours and either do another one as you repeat, or you can go home early, depending on what your day's looking like. So finally, we've got our detection. So at this point, you've got your membrane, you've got your protein of interest, you've got your primary antibody bound to your protein of interest, and then you've got your secondary antibody bound to your primary antibody, and that secondary antibody has either a HRP or a fluorescent molecule, so that you can then detect your initial protein of interest. So I'm going to go a little bit deeper into the chemiluminescent detection. So there are, like I said, you have a HRP molecule that is bound to your secondary antibody or your primary antibody if you use the flexible HRP. What you need to do is then add a substrate. Luminol is the substrate, which then gets oxidized by that HRP enzyme and releases light. Now, the substrate choice, there are options. You can have, so at Proteintech, we have Signal Bright Pro and we have Signal Bright Max. The Max allows you to detect up to femtogram levels of protein, so a really, really tiny amount of protein. So this is great if you're working with really low abundant samples, so if your protein of interest doesn't express very well, or if they're precious samples. So if you're working with patient samples and you don't want to use loads, you can use this really highly sensitive Signal Bright Max, which can essentially amplify that signal a bit further. So again, scan the QR code if you want a bit more information on the ancillary products that we've got for Western blotting. So now moving on to advanced tips. So I've been talking for quite a while. I've got about 10 minutes left. Hope you're still with me. So phosphorylation. So my advanced tips for phosphorylated proteins. So again, going back to my PhD, I was looking at KRAS, I was looking at MAP kinase signaling, I was looking at PAKT signaling. What I would always suggest for anybody looking at a phosphorylated protein is make sure you're aware of how many sites your protein can be phosphorylated at, and do those different sites, because a lot of the time your protein will be able to be phosphorylated at more than one site. Different sites will represent different biological context. So PAKT, for example, can be phosphorylated at serine 473 and threonine 308. And those two sites represent different activities of the cell. So make sure that you're aware of which ones you're interested in and which ones you should be targeting with the antibodies. In the lysate prep, include phosphatase inhibitors and protease inhibitors. You obviously don't want a phosphatase to come along and chop that phosphate group off your protein before you have a chance to detect it. Keep on ice, obviously, and then also enrich your sample. And by that, I mean load a bit more of your protein than you would usually, so that because essentially not all of your protein will be phosphorylated. You'll have maybe 15% of your protein will be phosphorylated. So you want to make sure you're giving yourself as much chance as possible to actually detect that phosphate group. So add more protein into your STS page. And also look for the non-phosphorylated version of your protein. So for example, in this case, we've looked at phospho-P53, and we've also looked at non-phosphorylated P53. So you can essentially look at the ratio of non-phosphorylated to phosphorylated protein, and that's always just useful information. If anybody's looking at small protein, so anything less than 20 kilodaltons, there are a few tips and tricks that we've got to make sure that you've got the most highly resolved Western. So using a tris trizene stacking gel instead of a tris glycine is useful. So this stacks your proteins more uniformly so that when it hits the resolving gel of the STS page, they'll resolve a lot neater. So as you can see on the right there. Overtransfer is a common mistake. So this is what I said earlier. Proteins can go through from your gel onto the membrane and then out the other side. So make sure that you're using the pore size that's right for your protein, so a small pore size, and also use semi-dry transfer. Don't use wet transfer for a small protein. And you can also soak your gel before transfer in an STS-free buffer, so probably DDH2O, before setting up the transfer, and that prevents the smaller proteins escaping the diaphragm or, again, over-transferring. Large protein tips. So again, the gel composition is useful to change if you're looking at anything above 100 kilodaltons. So higher resolution, you get higher resolution for large proteins if you use tris acetate gel rather than a tris glycine or a bis tris. This is because the gel is kind of floppier, so it's a bit less restrictive and it allows those larger molecules to migrate faster. If you're transferring onto a PVDF membrane, try and reduce the amount of methanol in the transfer buffer. Methanol and other alcohols can make large proteins precipitate out of solution. So reduce or remove as much as possible. And you can also increase the SDS detergent to 0.1% in the transfer buffer, again, just to make sure that those large proteins will stay in solution. For large proteins, I've also already mentioned this, but wet transfer is a bit more efficient. And you can also have a look at the higher molecular weight proteins in your protein ladder to make sure that they have transferred over. Western blot troubleshooting. A couple more slides and then we'll get on to the Q&A. So this view of a Western blot is most likely what most people have seen as their first iteration of Western. Usually you get extra bands. It's very normal. And that doesn't necessarily mean that you've got a bad antibody. There are a multitude of reasons why you might have these. So in terms of the higher molecular weight bands, these could actually indicate something that's going on biologically. So for example, glycosylation. So glycosylation is a post-translational modification which adds a sugar group onto your protein at specific sites on the amino acid code. And in theory, it should only add 10 kilodaltons, but actually what happens is that sugar group essentially reduces the ability of your protein to denature. So it will run a higher molecular weight than you're expecting. So actually higher molecular weight bands could be glycosylation. So you can add PNGase F into your lysis solution to cleave off these glycosylation groups if that's not what you're interested in. Phosphorylation can also make your protein run higher, and also higher order species. So dimerization, tetramerization, oligomerization, higher order species generation can cause these higher bands. So have a look to see if the higher molecular weight is a multiple of what you're expecting, and that would be higher order species. The lower molecular weight bands are kind of what you don't want. So this is indicative of proteolysis, so protein degradation, where your antibody is picking up your protein, but your protein has been essentially chopped up into smaller pieces. So it could be, something to do with the fact that you've got proteases still active in your sample. You've done multiple free store cycles, or you could have alternative splice variants of your protein. So there actually might be something that is biologically relevant. So just make sure that you're aware of what your protein can exist in. Isoforms, pre-proforms can exist. So for example, with insulin. So again, just make sure that you're aware of what states your protein can exist in before you kind of start changing your antibodies. So this is a troubleshooting summary. Essentially, if you've got high background, lower your antibody concentration, increase your blocking concentration and duration, titrate your primary antibody. So your supplier, the reagent supplier, should give you an initial starting point of what you should be diluting your primary antibody at, but do a titration for yourself in your own hands. Go lower, go a little bit higher, see what works for you. You can lower the exposure time if you've got high background, increase your washing time, and always optimize your lysis buffer as a priority. Your extra bands, like I said on the previous slide, use protease inhibitors and sonicate where appropriate. Use fresh sample where possible, and if you can't, make sure that when you've originally made your lysate, aliquot it into just single-use Eppendorfs. Understand your protein's post-translational modifications. So those extra bands might actually indicate that you've got post-translational modifications that you might not know about. Like I said, glycosylation can cause significant migration away from the expected molecular weight. And for lower signal strength, use a PVDF membrane, lower your blocking concentration and duration, check your antibody storage. So antibodies can go off, so just make sure that you're putting them in the right conditions for what your antibody needs. Load at least 30 micrograms or more of your protein if it's a new project and you've never worked with that before. Increase your primary antibody concentration and expose for longer time. So that's kind of the standard troubleshooting advice. I also used Able again for some other troubleshooting specific for KRAS, and it came up with some amazing examples of what you should look at if you are having issues. So if you do have issues, go back to Able, see if it can help you out. But in terms of the take-home messages from this talk, all the way back to the beginning, optimize your lysis and sample preparation as a priority. Your protein may act differently to a postdoc or somebody else in your lab group. So don't just use your protocol, their protocol, tailor your protocol for your protein of interest, and Able AI can help you out there. Don't just change your antibody if you have detection issues. There are multiple steps along the whole Western blot protocol that need to be optimized before that happens. And Westerns don't have to take forever. So Proteintech have developed the tools, like I said, which can speed up your Westerns and make your research progress a little bit faster. So this is my last slide. If you do want to learn more, we do have lots of stuff on our website. So lysate preparation, webinars, technical guides, blogs, and also for Western blotting. So we've got a protocol video, protocol guides, different blogs. And Poppy has been instrumental in actually developing a Western blot course, which is free and certified. It's on our learning portal, learn.ptglab.com. So if you want to get certified for Western blotting, that's also available to you. And thank you very much. I will hand over to Poppy now for any questions. Let me just try and stop my screen share. It's not working now. Oh, sorry, Poppy. It's all.
No, don't worry
There you
at all.
go.
That was really, really informative. Thank you so much for sharing.
No
We've had
problem.
lots of engagement on the chat, lots of questions come through, lots of people saying thank you for sharing your insights. So very clear and useful presentation. I will just quickly share one more slide with everyone and then we can get into the Q&A. So if there are any last minute questions, please, there's still time, please just put them in the chat. So I just wanted to share with everybody that all attendees for any of the talks within Immunoassay Week will receive a follow-up email, and that will include the recording of all of the webinars. You'll also receive individual certificates of attendance. Please do check your spam folder for those because they can end up there. And please do be patient. It can take up to 1 week for these to come through. And finally, you're all entitled to a 25% off one full-size primary antibody with the promo code antibody25. But again, all of this information will come through in the follow-up email. As Amy's already mentioned, we do have our online learning portal and lots of resources available on our website. And as well, there's 2 more talks this week at Immunoassay Applications Week. We have an immunoprecipitation workshop tomorrow, and then I'm going to be delivering a ELISA workshop on Friday. So if either of those topics are interesting to you and you haven't signed up yet, again, you can just find them on our website. If there's any other questions you have, you can get in touch directly with us at events@ptglab.com, or if you've got any Western blot specific questions, you can get in touch with Amy at amy.turner@ptglab.com. But again, this will all be shared with you in the follow-up email. So I'll just stop sharing my screen there. And as I say, it's been a very engaged audience, which is great to hear. We've got a lot of questions. But I'll start off with one that I think a lot of people have issues with is how can the overall workflow be adjusted to increase sensitivity for low abundance proteins?
Great question. So there's kind of a few things that you need to think about there. First is the clonality of your protein. So if you've got a lower of your antibody, sorry. So if you've got a low abundant protein, you want to amplify that signal as much as possible. So using a polyclonal antibody or a recombinant multiclonal antibody is kind of your go-to. Enrich your protein. You can enrich your protein, so you could even do an IP, pull down your protein and enrich it before you do the Western, but that might not work for all workflows. So if you can try and load a bit more protein onto your STS page blot. Use a PVDF membrane as well. So these have higher sensitivity, they have a higher capacity, so they will bind more protein than a nitrocellulose membrane will. And if you use a wet transfer instead of a semi-dry transfer, then that is more efficient. So if it basically make sure that all of that low abundant protein that is within your sample will move out of your gel and onto your transfer membrane. So it will just increase that detection essentially, or the chance that you will detect the protein of interest.
Great, thank you. And off the back of that answer, then we have also had a question about wet transfer. So you mentioned you can do it in as little as 1 or 2 hours, or it can be done overnight. What does the time depend on, and is overnight always going to be better?
Not necessarily. So the time depends on the voltage that you run it at. So the voltage is obviously the force that will pull your negatively charged proteins from your gel onto the membrane. If you use a lower voltage, it's going to move slightly slower. If you use a higher voltage, it's going to move faster. So you basically want to don't use a high voltage for a long period of time. Just make sure that it's dependent on that. What was the second part of the question? Is it always better? Not necessarily, but I think if you're looking at a low abundant protein, I think it is probably the best option for you. So it is more efficient to move proteins slower and longer. But this is because it kind of keeps them more happy. You're not putting too much kind of mechanistic strain on that protein, it could precipitate out. But if you're looking at kind of like a regularly expressed protein, you can just use semi-dry. I think that's absolutely fine.
Great, thank you. Some more questions as well about different experimental conditions. So someone has asked, when they're using a very highly concentrated protein lysate, so in this case it was a lyophilized lysate, do you have any suggestions to help minimize aggregation?
I would dilute it, honestly. Sometimes, I mean, proteins are tricky to work with. I always think of proteins as tiny little magnets. They've got charge on them, and if you've lyophilized a sample and it's really highly concentrated, you're essentially forcing these proteins to either stick together or crash out of solution. So I would dilute it to a point where they're not going to be kind of forced to crash out of solution. You can dilute it either in the buffer that it's already in or in the sample buffer. But yeah, ultimately try not kind of keep it really highly concentrated. You could potentially add detergents. It's like some detergents in there, so maybe STS, but I would basically have a look at the literature for the particular protein of interest. What else to keep stuff in solution? Yeah, bit of detergent, make sure the salt isn't too high. But ultimately, if you can dilute it slightly, that would probably be the best thing to do.
Great, thank you. So we've had a few questions come through about the blotting accelerator.
Yep.
So someone has asked, Actually, I might just run a few at you and then you can come to them in whatever order. So someone has
Okay.
asked, does the blotting accelerator work with antibodies from different companies? Someone has
Yes.
asked, yes.
It works for any antibody. Doesn't matter that it's from Proteintech or not.
Perfect. Somebody has said, how does it behave in terms of non-specificity? Because obviously you don't need to do the blocking step anymore.
So basically the kind of magic behind the solution is that it increases the binding kinetics between your protein of interest and the antibody of interest and the antibody that binds to your protein of interest. So it will increase those kinetic interactions, meaning that the non-specific interactions that might happen over a longer period of time are just reduced, essentially.
Great. And then final question on the blotting accelerator. How efficient is it when using to detect extremely low expressed proteins?
That's a good question. So what I would say is that if you've already optimized your antibody for your low expressed protein, it's kind of a substitute for the milk or the BSA or whatever that the blocking is that you're working with anyway. So it's kind of dependent on the antibody rather than the blotting accelerator. So if you do decide to switch to the blotting accelerator, you might have a bit of, you might have to slightly optimize it for the antibody of use, but no more than you would for kind of your normal milk or BSA.
Great, thank you. So one question which I think everyone who's done a Western blot before is going to relate to is, do you have any tips to avoid spillage to adjacent wells when you're loading a gel?
Make sure that the concentration of glycerol is high enough in your SDS sample buffer. So this is just going back to that slide, it increases the density of your sample. So it means that the actual sample will kind of sit lower than the running buffer. Try not to overfill, obviously. If you make your own gels, typically you'll get the little prong things. I can't remember what they're called. But the well bits of plastic that make the wells. And they come in like 10 wells or 15 well molds. So if you need to load lots of protein, if you need to load a larger volume because the concentration of your sample is too low, then I would always just try and use the 10 well ones, which have a little bit higher volume. And if you are really struggling, then just leave a gap between the wells. But you can also load the well and just leave it, let it settle, give it like, a minute, and then load the next one. And just try and be as careful as possible. You might, if you're using the Biorad cast gels as well, they can also, some of them come with like a mold that can help you actually load your gels, and they have like prongs, like a prong that sits on the top of the gel, and you can kind of add it like that if you're not super kind of well versed in loading gels a lot. What I would say is if you're doing a PhD with Western blots, by the end of your PhD, you're very, very good at it.
Yeah, unfortunately, I think that's one of those things that just takes a bit of practice, doesn't it? When you first getting your head around it. Well, good luck with your gel loading. Maybe just a couple more questions now because I am conscious of the time. But one person has just said, would you mind just repeating how to optimize gel percentage for the protein size?
Yes. So essentially, the lower the acrylamide percentage is best for higher molecular weight proteins. So this is because acrylamide is the component of if you make your own gels, acrylamide is the component of the gel that will basically give like the gel a mesh like structure. So it has pores in it. So if you're looking to resolve a really high molecular weight protein, you want those pores to be quite big so that the larger molecular weight proteins can resolve easily. So the less acrylamide you use, the bigger the pores. So that high molecular weight protein will migrate easier. If you're looking at a small molecular weight protein, you want tighter pores. So using a higher percentage acrylamide, like 15% typically, is better. So the smaller proteins will migrate through the small holes all the way down to the bottom of the gel, and the larger molecular weight proteins will remain at the top and migrate slower. So hopefully that answered your question. But our Western blot guide, which is on our website, will give you a little table for the acrylamide percentages that you should use and the relevant molecular weight proteins that might be of interest for that particular acrylamide percentage.
Great, very clear, thank you. And So we've had one question come through from someone who's using a semi-dry transfer method.
Mhm.
They've said that they are seeing their bands at the correct size, but the bands often aren't complete. So they're either only getting half their bands across or it's just not very reproducible. Do you
Okay.
either know why or any advice on how to stop this?
So there's a couple of things that it could be. Either the transfer, the pressure on the plates is not uniform. So you need to make sure that when you're making that stack, the gel membrane blotting stack, that there's uniform pressure on each side. So maybe just try using a different cassette if it's that, and make sure that you roll out the bubbles. Or it could actually be to do with if you're using chemiluminescent detection right at the end of your protocol, you need to make sure that the chemiluminescent detection substrate is uniformly distributed across the whole bands. Because if you've only covered half of the band with the ECL, then you're only going to have the HRP reaction for half the band. So you've either got a transfer issue there where the pressure isn't consistent across the whole plate, or it's a ECL issue, a chemiluminescent substrate issue.
Great. Okay, one final question. Being an antibody company, we need to answer the antibody question.
Mhm.
Someone has said that they're using a polyclonal antibody through their Westerns, and they're not seeing the same answer every time. So they don't think it's an antibody issue because sometimes it detects for Western, sometimes it doesn't. But even when it doesn't, they are seeing the loading control. So they don't think it's a problem with the Western. Do you have any reasons on what that could be or how they can improve reproducibility with an antibody?
So if the controls are all working, then it could be an antibody issue. Polyclonal antibodies, what I would suggest is that you use it fresh each time. So quite often people will, use their antibody, put it in the milk or BSA, use it once, put it in the fridge, use it again. Don't do that if you're seeing, especially if you're seeing reproducibility issues. Make sure that the lysates are also fresh, because what it could be is that your actual protein of interest might be degrading. So make sure you're using fresh lysates. If it's a polyclonal antibody, if you're using the same lot and you're still seeing different issues, then I don't think necessarily that it's an antibody issue if it works sometimes, other times. But make sure that you're storing the antibody correctly and make sure that you're not reusing the same antibody from like when it's been diluted. You could also consider, obviously, if there is a recombinant version of your antibody, trying that, because if the reproducibility issue is between different lots of your polyclonal antibody, then the different type of clone that has the clone of an antibody that has the highest chance that there is variability between the lots is a polyclonal. So if there is a recombinant version, use that. At Proteintech, they're exactly the same price, so there isn't like a premium on that. So try that, and it might work for your issues.
Great. Thank you so much, Amy. I think we are going to have to call it there, as I say, just because I'm aware that you'll all have experiments to be getting back to. If there's any questions that we haven't got around to today that you do want us to answer, as I say, please get in touch with us directly, or you can look on our website. Amy's already highlighted all the different resources we have available to help with Western optimization. But I just want to say a huge thank you to everyone for attending today. Massive thank you to Amy for sharing your Western blot expertise. I hope you all have a wonderful rest of the day, and I will bring the webinar to an end.
Thanks, everyone.